All right, thank you for another week of Soul Seminar Online. Today I have the honor of introducing Carmen Fernandez. She's a Peruvian biologist who graduated from the National University of San Agustín and holds a master degree at Taxonomy and Biodiversity of the Imperial College London, thanks to the Chevron Scholarship System. Currently, she is working on the floral specialist of the National Forestry Services and Wildlife of Senfron in the city of Monzcal, Peru, and she's a researcher at the Institute of Scientific Michael Owens Dillon in the Herbarium of Sir Pruviano in the city of Arequipa, Peru. So with that, you can go ahead and start sharing your screen and start when you're ready.
Okay, thank you, Chelsea. Hi, everyone. As Chelsea told you, my name is Carmen Fernandez, and today I'm going to give you this brief talk. First of all, I'm a bit nervous, so please bear with me if I make any mistake with my English. Well, right now I'm working as a researcher at the Herbario Sur Peruano in Arequipa. And in this case, I'm going to give you a talk about the genus Leptoglossis. This is a pretty small genus, and this is a project that I did for my master thesis with the help of Sandy and Tina. So I decided to title this talk, A Tiny Genus of Argentina and Peru, Leptoglossis. First, I'm going to give you a brief description of the genus, then I'm going to talk about what's going on with its taxonomy and what are the main reasons that I decided to conduct this kind of study. So as you can see in these pictures, the genus Leptoglossis, all the species are annual or perennial herbs. They're pretty tiny. Most of them are between 10 centimeters to maybe 30 centimeters tall. And they have these tubular and zygomorphic corollas. They vary from white to yellow, and with only one exception, that is Leptoglossis acutiloba, who has these purple veins in all the corolla. And other thing about this is that this species, we're only going to find it in very specific environment. So as you can see in this picture, they inhabit these very xerophytic, arid environments in Argentina and in Peru. And maybe it's going to be a bit difficult to distinguish the species among all these other species who are here in this sandy soil. But here, exactly where I'm pointing right now, is one individual of Leptoglossis. Here is another one, and here another one. So these species usually inhabit this kind of environment. And for example, this picture is from Arequipa, and we're going to find this species, the Leptoglossis acutiloba, living with other species of Boraginaceae, for example, Tichilia. And here in the bottom right, there's another genus of Solanaceae, Exodeconus. So this is the kind of environment that we're going to find this species. But sometimes when there's a very humid season, we're going to find also the species of Leptoglossis in this kind of slopes. Usually slopes with sandy and rocky soils. And even if you cannot see it clearly, all this slope is covered with Leptoglossis. But in this case, with another species of Leptoglossis, this species is Leptoglossis albiflora. And if we check its name, this species has white flowers. So maybe it's one of the reasons that it's pretty difficult to try to distinguish the species among the other ones. But also we're going to find Leptoglossis in these other kind of environments. I don't know exactly the translation in English. I call it quebrada seca. And it's these places where in one season is going to be water, maybe in a few years, it's not going to be any kind of water in here. So the species of Leptoglossis are usually going to be here in this bank of sand, or sometimes near these rocks. But usually almost all the species of Leptoglossis are going to live in this kind of environments. Another thing that I would like to say in this introduction about the genus is that Leptoglossis, according to the characteristics of Hunz and Subils, they say that there are 7 species with a disjunct distribution. 1 species in Argentina, and the other that is Leptoglossis linifolia, and 6 species in Peru, that is acutiloba, albiflora, darcyana, ferreyrae, lomana, and schwenkioides. Also, something that I would like to highlight is that the species in Peru, they also have a disjunct distribution. There are species who are going to be found in the lomas formations, for example, lomana, ferreyrae, and darcyana, especially southern Peru. And the other species are going to have a more widespread distribution in northern Peru and in central and southern Peru. So they also have a disjunct distribution. So that's a brief introduction about characteristics of the genus and the distribution of it. But at the same time, I would like to talk about what's going on with its taxonomy. Well, the genus was first described by Benth in 1845, but when he described this genus, he said that it was part of a tribe, Salpiglossidae, and he said that it was part of the family Scrofulariaceae. However, 4 years later, Miers did a review of this tribe, and he said that this tribe, in fact, it wasn't Scrofulariaceae, but it was Solanaceae. In that time, the genus Leptoglossis was in the tribe Salpiglossidae with the genus Salpiglossis, Browalia, and Pteroglossis that right now is Regesia. However, the relationships of Leptoglossis with other genera has been always changing, and it depends on the criteria of each one of the researchers. So, for instance, Weinstein, he said that Leptoglossis, it was only a section of a bigger genera, a bigger genus, that it was Salpiglossis. But years later, Hunziker, he said, no, in fact, Leptoglossis, it's a genus, it's not part of Salpiglossis. Also, he said that Salpiglossis was part of another tribe, Salpiglossidae, and that Leptoglossis was part of the subtribe Incestroidae. The most recently analysis and revision of the genus is the one who has been done by Hunz and Subils in 1979. And something that I would like to say is that this kind of revision was done using only morphology data and herbarium specimens. And in this kind of analysis, they say that there are 7 species, the ones that I told you before, 6 in Peru and 1 in Argentina. More recently, molecular analysis, they have concluded that Leptoglossis is in fact part of the clade Pétunier, but I would like to highlight these two, that the only species who have been sequenced until that time were Leptoglossis linifolia and Leptoglossis darcyana. So in fact, we don't have data for acutiloba, albiflora, ferreyrae, lomana, and schwenkioides. So with all that in mind, I said, okay, I'm going to do this kind of project, and my main aims are going to be these two. First, I would like to delineate the genus Leptoglossis using morphological and molecular data to try to help understand the relationships within the genus, in particular in regard to this young distribution. And also, I would like to test whether that is young Leptoglossis linifolia is a member of the genus or is more closely related to other taxa. So for this, I use plant material in my methods, and I decided, okay, I'm going to conduct this study using 2 kinds of analysis. One is the morphology analysis, and the other one is going to be the molecular analysis. But for using the morphology analysis, I used the following plant material, that it was 184 specimens. These specimens were physical material and the other were scans that I downloaded from Global JSTOR plants. And this material was from this herbarium that you can see here, the Natural History Museum, Core, Edinburgh, Field Museum, other Kew, New York, and herbariums also in Peru, like Herbarium Truxiensis, Herbario Sur Peruano, and the University of San Marcos. And for the molecular analysis, I used 22 specimens from the Natural History Museum, Core, Edinburgh, and Herbario Sur Peruano, and San Marcos, and 15 accessions from GenBank. For the purposes of this talk, I'm going to start with a morphology analysis. I'm going to explain my results of the morphology analysis, and later on, I will talk about the molecular analysis. And with that, I will start. So when we are trying to describe new species, or we're trying to do the taxonomy of one specific group, we always have in mind that we have different kind of characters that are going to represent the shape of the plants. And those characters usually are qualitative characters, for example, the kind of pubescence that we're going to find in the surface of the leaves or the stems. But at the same time, we're going to find quantitative characters. And those characters usually are measurements of the leaves, the stems, the size of the flowers, or we can use ratios. And these ratios sometimes represent better the shape of a structure. So with those characters, I say, okay, I'm going to try to conduct a specific kind of analysis. And this analysis is a multiple factor analysis. If you ask me why I decided to use this kind of analysis, it's because at the end of this analysis, we're going to have a graphic that is going to represent a morphospace. So if we have a dot for each one of the specimens that I measure and took their qualitative characters, we're going to see in the morphospace all the specimens who are most close related to each one are going to be clustered in one group, and the other specimens who differ from this are going to be clustered in another group. So we're going to see everything in a morphospace, and maybe we can infer if they're like different species or not. So it's similar to a principal components analysis. So first, when I did this multi-factor analysis, I said, okay, we're going to have all these characters. I use 45 characters who are grouped in qualitative characters like present or absence or a rosette, the color of a corolla, if they have or not have purple veins, the number of the calyx lobes. For the quantitative characters, I use the measurements, as you can see here, the length of the flowers, the leaves, or the width of the throat, the length of the pedicel, and things like that. And finally, the radius, the radius like the length and the width of the flower leaves, cauline leaves, the calyx, and the total length of the flower in relation with the length of the pedicel. So if we're trying to represent all these characters in a multiple factor analysis, there are going to be some characters, in this case, we're going to call it variables, who are going to give a more contribution to this morphospace. For that reason, I decided to do the first analysis, that is a multi-factor analysis, who allow me to identify the characters with the highest variation within the genus. And later on, since I know which are the characters with the highest contribution, I found that the characters with the higher contribution to describe this genus are the quantitative characters, but also not the measurements, but the ratio. So, for example, I have the results of my morphology analysis, and I found that I can express in a morphospace with 2 dimensions, and these 2 dimensions be the qualitative characters, but also are going to be the shape of the flowers and the shape of the leaves. So if we remember that, we're going to have this. This is the morphospace, and in this morphospace, in this graphic, we're going to find, this graphic is going to represent the correlation between the variables and are represented in each dimension. So for instance, if we find a dot here, and this dot is going to represent one of the specimens that I measured, I'm going to say that this specimen is going to have a calyx that is more similar to this one. But if I found a dot that represents another specimen here in the top right of the graphic, the calyx is going to be more closely related to this kind of shape. And the same is going to happen with the lips. If I found specimens in this part of the graphic of the morphospace, the lips are going to be more similar to this one. They have wider lips in the top left of the graphic. So I have this kind of morphospace, but at the same time, the MFA analysis allowed me to check what are the qualitative characters who are giving more contribution to distinguish among all these species of the genus.
Carmen, I think you froze.
The stems, something that I would like to.
Can you go back a couple? You just froze.
Of trichomes, whereas in albiflora, we're going to find glandular trichomes, the same that is going to be in schwenkioides and in linifolia. But if we're going to check what's going on with the species who inhabits the lomas formations, like darcyana, ferreyrae, and lomana, we're going to see that they do have branching trichomes on the stems. So with this information, at least with the MFA analysis, I can start seeing what are the characters who are going to be more important to try to differentiate between all those species. Also, another character that is pretty important is the number of lobes in the calyx. So with all of this, this is the morphospace that I found in this analysis, and I found something that may be surprising. And this is that in this side of the morphospace that we find the leaves who are wider, we find two species. That is the Argentinian Leptoglossis linifolia, but also the Peruvian species Leptoglossis. Both of these species, they share some characteristics, the morphological characteristics, but both of them are pretty good represented in two. But if we're talking about the other Peruvian species, we're going to find a mix. And this mix, for example, is here with acutiloba and albiflora. Even when both of them are two clear groups, we see that there are some characters that are overlapping. But if we take into account another quantitative characters, like for example, the color of the corollas, the number of lobes in the calyx, and also the presence or absence of glandular trichomes in the stems, it's pretty easy to identify both of these species. What I found surprising is what's happening with the species in the lomas formations. And the species in the lomas formations, there are three: Leptoglossis darcyana, lomana, and ferreyrae. Leptoglossis darcyana is isolated in only one location in southern Peru, in the city of Tacna. I couldn't find this species in any other locality. So right now it's endemic of this locality, darcyana, and darcyana is like It's in the middle of the characteristics that we're going to find in acutiloba and albiflora, but at the same time shares some characteristics with the other two species in lomas. This lomana ferreyrae has been a matter of discussion with Professor Victor about if they do in fact are one species or two different species, and I will talk about this later. But the results of my morphology analysis is that they are the same group, they share the same characteristics. So using only morphology and only the characters that I explain here, I can say that those two species are the same thing. So with that, I would like to say that lomana and ferreyrae should be the same species, at least using this analysis. Linifolia and schwenkioides, they share some similar characters, but are pretty different from the other Peruvian species. And the other species that we can find is acutiloba, darcyana, and albiflora. So that's all about my morphology analysis. And the other thing that I did was a molecular analysis. With this molecular analysis, as you can see, here is the workflow. And I did a PCR amplification, and I used 4 chloroplast regions, that is PSVA, TRNH, TRNLF, TRNSG, NDHF, and I use 1 nuclear ribosomal ITS2. And for the outgroups, I use Nierembergia, Bauketia, and Petunia axillaris. And I did a maximal likelihood and a Bayesian analysis. With that, I found that there was some kind of incongruence between the phylogenetic tree that I obtained from the chloroplast and the ITS. So I'm going to present you these 2 different trees, what I found. First, I found 2 clades, and it says that in fact, Leptoglossis is monophyletic. I found a clade of the Argentinian species, linifolia, and I found that the other species in Peru found a pretty clear clade. And also I found that it's easy to distinguish schwenkioides, but schwenkioides, I think that maybe it's an early split from Leptoglossis linifolia, and it helped me to try to understand what's going on with the morphology, because if we remember what was going on with the morphology, these two were really, really close related similarities when morphological characteristics. However, I couldn't find what's going on with the relationships within the other species. I couldn't find like a well-supported clade with acutiloba, albiflora, lomana, darcyana, and ferreyrae. In fact, I found that sometimes acutiloba was close related with albiflora, but at the same time, I found another clade with albiflora and ferreyrae. I think that maybe this is represented because when I tried to, when I did the PCR amplifications, I couldn't find, I couldn't amplify all the genes used here. And maybe that's one of the reasons. Also, we need to remember that I tried to use herbarium material, and sometimes it's difficult to try to extract DNA from this kind of material, especially if this material is pretty old. When I talk about a nuclear ribosomal DNA ITS, I found that there was a difference of this one, that I found 2 clades, the Argentinian clade with linifolia and the Peruvian clade. Here I found a difference that albiflora and schwenkioides, who are the 2 species who have a more widespread distribution in Peru, are mixed with Leptoglossis linifolia. So maybe they can give me some kind of conclusions. Is concerted evolution, maybe this is representing the history of the gene, and know the history of the species. By the time, it gives me another hypothesis that maybe the Leptoglossis linifolia is the first species in the whole genus, the ancestral one, and maybe it has a widespread distribution in South America, and later from different kinds of speciation, it became schwenkioides, that is northern Peru, and later albiflora, that is in central and southern Peru. And these species like darcyana and lomana and the species that we're going to find in the Lomas Formation are new species because they're following the same patterns of the other species that we're going to find in the Lomas Formations. So with my discussion, I found the following. Leptoglossis is monophyletic. There are two clades, the Argentinian clade with Leptoglossis linifolia and the Peruvian clade. And there are three species who are distinct based in morphology and molecular data. Those ones are schwenkioides, that is the one represented in blue in northern Peru. Darcyana, the one represented in yellow in southern Peru in this locality in Tacna. And these are the three species that we're going to find that are pretty distinct. Linifolia, schwenkioides, and darcyana in yellow here. Also, even when the molecular analysis, I couldn't find like a real clade and separation between these species. If we focus only on morphology, we can say that these are two different species. And this one, Leptoglossis albiflora, who is one of the species with widespread distribution in Peru, that is represented in orange. That is this one with white flowers, sometimes yellow flowers, and this kind of calyx with more than 5 lobes. And the main characters to distinguish both of these species is the calyx and the type of trichomes. In Leptoglossis albiflora is the species who has more glandular trichomes than Leptoglossis acutiloba. And Leptoglossis acutiloba is another species with widespread distribution, but only in southern Peru. We're not going to find this species in northern Peru or central Peru, but only southern Peru, but in different cities. And both of these species are going to be in medium altitudes from 1,000 to 3,000 meters above the sea. Another thing that I would like to highlight is that in Hunziker revision, he said that Leptoglossis acutiloba lacks of a basal rosette. But if we look in the field, we find that in fact they have a rosette. And the same thing is happening with Leptoglossis albiflora here in the left. They do have a basal rosette. Maybe the conclusions that he gave in his revision is because he checked only herbarium material. And sometimes when these plants are in an older stage, they tend to lose this rosette. And as you can see here in the pictures, another difference is the pubescence in the stems. Here in Leptoglossis albiflora and here in Leptoglossis acutiloba. This is one topic of discussion with Professor Victor, and is if we should treat these two species as a single taxon or as two taxons. From my point of view, I think that they should be treated as a single taxon based on the morphological analysis, and also based that when we have all the specimens who have been collected in Peru, because these two species are only going to be found in one city, in Arequipa, but in different localities in Lomas Formations, is that the specimens who are in southern Arequipa, in this city, are going to be more like this picture. But the specimens who are going to be found in northern Arequipa are going to be more similar to these ones. We can see some kind of difference in the inflorescence. But if we check the other species of Leptoglossis, we can see that sometimes this kind of arrangement of the flowers depends on the quantity of water that they do have. At the same time, all the species of Leptoglossis ferreyrae has been collected during the Niño events when there's a lot of rain and water there's a lot of water in the lomas formation. So maybe this kind of conditions have allowed these kind of characteristics in the plants. Also, I'd like to say that if we check the prologue of Hunz when he describes Leptoglossis ferreyrae, he says that Leptoglossis ferreyrae lacks one of the main characters to differentiate from Leptoglossis lomana and Leptoglossis ferreyrae is that Leptoglossis ferreyrae lacks a basal rosette, but in fact they do have a basal rosette, but I don't think that this is enough character to differentiate both of them. So that's one of the reasons that I suggest to treat these two species as a single taxon. And since Leptoglossis lomana was the first described, the name that should be called from now on this species should be Leptoglossis lomana. So I conclude the following: the monophyly of Leptoglossis. There are two clades, the Peruvian species and the Argentinian species. There are three species who are distinct in morphology and molecular data, that is darcyana, linifolia, and schwenkioides. Two species should be treated as distinct basic only on morphology, that is albiflora and acutiloba, and two species should be treated as a single taxon, that is ferreyrae and lomana. I would like to say that maybe in the future we need to use more nuclear markers like waxy on Lefi. I couldn't use this kind of markers. I tried to use waxy, but the primers weren't working, at least the ones who were used by Levin in 2006. So maybe we need to redesign new primers, at least to study the species in the lomas formations. And we need more accessions of Leptoglossis. I only used 22 specimens, but of these 22 specimens, very few, I couldn't obtain all the markers from all those specimens. Also, the other thing is that I only use characters like the shape of the corolla, shape of the calyx, but I didn't use reproductive structures, and I didn't use characters of the fruits because it was going to be difficult to obtain this kind of data from scans found in JSOR global plants. So this is my presentation, and I would like to say thanks to first Sandy and Tina, who helped me during my project for my master thesis, Dr. Gloria, Michael Dillon, and Tina, because they provide me with the plant material for the DNA extraction. Stephen Russell and Tina helped me with the lab work and the phylogenetic analysis, and also Daniel Montesinos, Italo Trevino, and Victor, because they allow me to use their photographs for the plates. And that's all. And I hope that you like the presentation. Many thanks, and any questions I would like to answer, I don't know. Thank you.
All right. Yeah, thank you so much, Carmen. I think taxonomy work is really important, and it's great that you're getting into this detail and looking at different stuff. So if you have questions, you can type them in the chat, or you can just mention that you have a question and we can call on you. Nice. So going through this, it seems like there's still small difference. What's your opinion on like subspecies? Do you think those would be helpful in these scenarios? Do you think like there's going to be a big genetic differences between these two? When you're like grouping it into one species, do you still think there's a little bit of a difference between those populations that could be distinguished, or you're just like, just call it one species, keep it simple?
I think that it would be better to keep it as a one single taxon, as Leptoglossis lomana, because when I was checking all the material, as I said before, it's pretty difficult to to make a line when all these species are going to be Leptoglossis ferreyrae and Leptoglossis lomana. So since also these species are found only in the lomas formations, and in the lomas formation, there's a lot of speciation, I think that maybe this species could be a subspecies, a variation, or maybe it's just beginning an speciation process. But I don't think that is conclusive to say that there are two different species. So I prefer to treat it as a single taxon.
Sandy had a comment. I don't know if she wants to.
Yeah, so Carmen, that was fantastic. That was a brilliant talk. I think your observation about ferreyrae, about Leptoglossis ferreyrae, that it is only collected in the El Nino years, is really interesting, because that's something that tells you, and those plants in the lomas, of course, are so variable based on the environment. I mean, I know that's true with tomatoes. And so you could even do experiments with lomana and grow it under high water regimes and low water regimes and see if you can recover the different morphologies. That would be really amazing.
So I think that would be a fantastic project, because I have never done something like that. And I think that it will allow you to understand more about what's happening with this kind of plants, because it's the same. This at least Leptoglossis ferreyrae has only been collected during the Niño events. So maybe it's because of the availability of water, and is we're seeing just phenology, but not another characteristic.
Yeah, that's great. Really great observation.
All right, I think Stacy has a question now.
If my video is working this morning, it's working.
Yep.
That was so awesome. I have to confess to you that I have spent an embarrassing amount of time looking for Leptoglossis and failed consistently. Once I saw them with Segundo in northern Peru, but other than that, on my own, I have entirely failed. And so one thing I've wondered, just to make myself feel better, is, but also sad though, if this were true, do you think any of them aren't there anymore? Like, there are all these little species sprinkled across the lomas, and some of the collections were super old, and I couldn't find any recent collections. And now I don't remember which ones were missing recent collections, but I wondered, like, did any of them just disappear between, you know, the 70s and now?
I think that maybe with Leptoglossis, well, I think this is one of the reasons that I differ with Professor Victor, because he said that there's in fact Leptoglossis ferreyrae. But when I did my undergrad thesis in 2014, and since nowadays, I couldn't find any of those species on the field. So it's one of the other reasons that I think that maybe everything is Leptoglossis lomana and not Leptoglossis ferreyrae. About schwenkioides, yes, there have been recent collections about Leptoglossis schwenkioides. And I think the other reason that there are not many collections about this species, and most of them are pretty old, is that it's a bit difficult to find them on the field. As I showed you in the first slides, when you go in the field, since they're really, really tiny, it's really difficult to find them. Something that maybe helped me to find at least Leptoglossis acutiloba is that when you see these species from very far away, some of them resemble tagetes from Asteraceae. So because of the color of the purple corollas. But I think that maybe I don't think that any of those species has been lost, but I think they're endangered, especially the species who are in the lomas formation, like Leptoglossis darcyana. The only locality who has been found is in Tacna. And I think the last collection has been like 10 years ago. And I haven't been able to visit this location, so I don't know what's going on with that species. The other thing that gives me many questions is maybe there are any other many other species in Chile. I don't know, because this is young distribution in Argentina and Peru. Maybe there's something in between, but I haven't collected there, and there are not any collections then, so I don't know. Maybe there's another species in this environment, or they're like some kind of barrier I don't know. There
Yeah,
are many questions.
it would be super interesting to find that out. Like, I'm very fascinated by this genus. One other thing I was wondering about, see, because I haven't gotten to see these things in the field, is their little stems looked so sturdy. I wondered, do you think they're entirely annual, or do you think some of them manage to make it through a season, especially the ones that are more inland, where maybe it's, well, I don't know, maybe inland's just as dry and harsh as the coast?
I think maybe with schwenkioides, I'm not really sure. And maybe with linifolia, but with the other species, I think that of them are annual, because I've been on the field in months of the year trying to find these species, because not all the years is possible to find them. So maybe with the schwenkioides and linifolia, I haven't seen those on the field, but from the characteristics of them, I think that maybe they could be perennial if you give like enough water to grow them. But with the others, no, I think they're annual.
Okay, I have one last question. It's funny, schwenkioides and linifolia are the only ones I have seen in the field. So you and I have seen unmatched groups, like you've seen all the ones I have. But so in Argentina, linifolia is really variable. And Franco, who I don't know is here today, Franco really thinks there's 2 species. What do you think about that? Do you think there might be multiple species in Argentina? Or do you think it's a continuum, really?
Sure about the Argentinian species, because I only seen the herbarium material, but I found these different papers from Cordoba where I say they have pink flowers, white flowers, and I think this, I'm not really sure if they could be like different species. Maybe it's something that is happening with Leptoglossis acutiloba, because in Leptoglossis acutiloba I found some specimens with yellow flowers, other who looked more like purple flowers, but but the most of them, they're yellow with purple stripes. So I don't really know what other characteristics have Leptoglossis linifolia to say that there are different species, because I only checked the herbarium material. I haven't seen any of them on the field, but I think that it would be really interesting to check those species on the field and maybe do more collections and a different kind of molecular analysis with all of them.
Yeah, well, I'll get Franco to send you some pictures, because they do look quite different in addition to the flower color, but I don't remember. Anyway, other people have questions, but I'll send you an email.
Yeah.
I think Sandy had a comment about what Stacey was saying too.
Don't worry about it. There's a question from Alfonso Orellana. He's wondering if you could have Leptoglossis schwenkioides in southern Peru, and please answer in Spanish.
Okay, hola Alfonso, en base a lo que yo he revisado y las colectas que hay, yo no creo que haya schwenkioides hacia el sur de Perú, únicamente hacia el norte. Lo que a mí me parece más interesante es el tipo que creo que deberíamos buscar un poco más acerca de la historia de este género, porque mi hipótesis es que quizás quizás Leptoglossis linifolia fue la base de todo Leptoglossis y que tuvo una amplia distribución en toda Sudamérica, y que con los diferentes periodos y cambios que ha habido en Perú, schwenkioides sería como la primer divergencia del género. Y que luego de schwenkioides ya se presentan las especies que las encontramos a lo largo de los Andes, como acutiloba y albiflora, que son las que tienen una amplia distribución en Perú. Entonces, yo creo que ya se dio un cambio, como un corte con schwenkioides, y que quizás lo único que encontremos hacia el sur de Perú sea acutiloba y albiflora, y que las especies más recientes serían las especies de lomas.
All right, thank you. Let's see, is there any other questions or discussion that we would like to have for Carmen?
No.
Well, you did great. Thank you so much. Next week, Janet Sullivan is going to be giving a talk on what's next for Physalis. So hopefully hopefully you can make it. Thank you so much, Carmen. It was great. I'm sure she's available by email if you have more questions. All right, have a great weekend, everyone.
Thank you so much. And anything, just write me. Bye, and take care, please.